Kind of do like a little matching. I've got a whole bunch of random, unorganized words here. Nucleus, amino acid, DNA. And so what do you think this is up here?
The nuclear membrane.
Alright, so the little blue thing is the nuclear membrane. So what should be inside the nuclear membrane?
The nucleus.
Yeah. Your DNA.
The DNA should be inside the nucleus. Okay, so, alright, so don't yell out, just kind of, let's see if you can follow this. Like I said, you need a codon chart to help build this thing. Alright, so over here, on the wall over here, follow me, I've got some DNA, I've got some RNA. So let's do this. By the way, CTY, or CYT? What? Cytoplasm, abbreviation for that. Okay, so, DNA, right? So I should be able to write this strand. What is that strand called? If it's another DNA.
Double helix.
Alright, the double helix. Okay. Alright, so let's see. Got my DNA label. I got my nucleus.
No.
Nucleus. Yeah, the DNA. I'm going to actually put the DNA over there. Shh, people taking a test. Hey, that would be you as soon as we're done with this. So, alright, so what is connecting these?
Hydrogen bond.
Alright, raise your hand. Hydrogen bond that connects the bases. So what is not shown in this model? The parts of the DNA molecule. Someone from over here. What do they think about the three parts of a nucleotide? If this is the rungs of the ladder, what are we not seeing?
The sides.
The sides. The sugars and phosphates. Sugars and phosphates. Okay, now remember, DNA is replicated. DNA is transcribed. DNA is replicated. So if we were to replicate it or transcribe it, the first thing we've got to do is do what?
Unzip. You have to replicate.
Unzip it. That means we've got to break these hydrogen bonds. We have an enzyme called helicase. Remember, helicase unzips and it kind of unravels the DNA so that we can get more DNA here, more DNA here, if this was replication. Now, we make a copy of the DNA so that we can do mitosis. Remember, the chromatids separate into two cells? Now, we'll do transcription. So let's do this. We just unzipped. By the way, there is an enzyme that would have added the free nucleotides of DNA. What's that called? Helicase opens up. Free nucleotides come in. Free nucleotides, if you're making more DNA, or if you're making RNA, the same ending. Polymerase.
Oh, yeah.
All right, some of you are nodding. All right, I should have let you give me the answer. So if this was a replication, it'd be DNA polymerase. But now, we're going to do DNA transcription, which is DNA making messenger RNA. So, see, it should pair up, right? Now, I got DNA there. Let's do this. As soon as the sugars and the phosphates on the sides form, hydrogen bonds temporarily connect it, so it kind of holds it into place. Now, let's unzip, break that. Let's do this, kind of flip. What am I doing now?
You're
zipping it back, reforming the hydrogen bonds. Good. Okay, so boom. There we are. We got our messenger RNA. That's transcription. Now let's do this. Imagine a little hole in this nuclear membrane. Okay. Save a little space. I'm going to do this. I'm going to move the DNA from the nucleus over here. And watch this. Hey, what's that little bubble?
Ribosome.
Ribosome. So I can grab some more things. I'm going to put the mRNA there. And I'm missing a card. There's your ribosome. Actually, and this is the one that students keep getting confused by. That's the rRNA. It takes all three. The RNA makes up the actual structure that is the ribosome. So now let's do some, okay, wait a minute. mRNA, rRNA, what are we missing?
tRNA.
Okay, tRNA. Let's get some tRNA up here. Let's do this. Just like that. There's tRNA. And it's going to have an amino acid on one side. And this bottom part, what's that called?
Codon.
If it's tRNA versus RNA, what are your choices?
And
Codon.
Anticodon. So I'm going to go over here and I'm going to label the codon, and I'm going to put the anticodon over here because see, anticodon bonds with the codon. So tell me, AUG is going to be now this is, don't do this on the test though. If I give you DNA, write the RNA, and then use your codon chart to tell me what the tRNA is going to carry in. So let's do this. That's our tRNA. Tell me what amino acid is going to be carried in to hook up with AUG.
Methionine.
Okay? Remember, you use your codon chart. First base, second base, third base. So here's methionine. So here's our first amino acid. Boom. Okay. So now, okay, this is going to be our next one. Who's got a codon chart? Tell me what's going to happen next. Huh? GGC. Glycine. Okay, now watch this. And the next step is actually going to be the part that's, it's an overly simplistic visual, but, all right, let's see. Let's make sure this is, okay. Now, before I bring in the next amino acid, why don't you tell me, what would be the next amino acid? AGU. What? Serine. Now watch this. Before it comes in, I'm taking that off. And now this, as the ribosome, imagine the ribosome's moving down this direction. So there, this floats off. Does that make sense?
Yeah.
This is what translation is doing. DNA is transcribed, RNA is translating. The ribosome is translating the amino acid that will be produced. So let's do the last one. Make sure that they pair up, okay? What is it? That means it's GUC. Valine, okay. So let's do it like this. I move these over out of the way now. Okay, pull that off because we got one more codon here. What's this?
Stop.
It's a stop codon. So that says, hey, we're done. Get this out of the way. We're done. What do these do? They go pick up the next amino acid for the next time an amino acid chain is needed. Okay, so let's pull this over here and let's fold it like it's a functional protein or something. Okay, now let's add some labeling. Okay, that's an amino acid. The whole thing is a protein, and the little yellow bond in between is a peptide bond. And that's how we go from DNA, follow me,
Okay.
DNA, being transcribed in messenger RNA to be translated to produce a functional protein.
Okay.
Are there any questions?
No.
Good, Kat.
I know.